I have been following the work of embryologist Marcelo Barbieri since 2000. In August of that year, I came across the Portuguese translation of his 1985 book, The Semantic Theory of Evolution. An amazing book. I read and reread several times and contacted the author later on. Since then, we have been keeping in touch. Marcelo Barbieri has gradually revolutionized contemporary biology. As a curiosity, both Karl Popper and René Thom, René prefaced the 1985 book, have placed Marcelo as a milestone to take into account. And more recently, Gazzaniga, in his 2019 book, The Consciousness Instinct, points Marcelo as a real name for understanding the phenomenon of life and consciousness. His proposal is called code biology. Code biology is the study of all codes of life with the standard methods of science. The genetic code and the codes of culture have been known for a long time and represent the starting point, the historical foundation of code biology. What is new in this field is the study of all codes that came after the genetic code and before the codes of culture. Some of those new codes have already been discovered and many more will likely come to light. The existence of many organic codes in nature is not only a central experimental fact, it is one of those facts that have tremendous theoretical implications. Since 2003, I have been trying to bridge the line between code biology and psychology, and later between analytical psychology and its archetype concept. Interesting parallels exist between archetypes, understood as ancient forms of behavior typified in memories of the species, and the idea of neuronal codes. I believe archetypes are codified in neuronal codes, something to be proved in future neuroscientific experiments. But now, let me present you with the thing that matters. We went to Ferrara, Italy, where Marcelo welcomed us into his house. We carry in our luggage a series of questions. Marcelo answered us in the form of 4 grand statements.
You asked me about code biology, and it is important to begin by saying that code biology is 2 very different things at the same time. First of all, it is a new field of experimental research. More precisely, it is a study of all codes that exist in nature, from the genetic code to the codes of culture. At the same time, code biology is a new theoretical framework, a new paradigm. Today, there are 2 dominant paradigms in biology. One is a chemical paradigm, the idea that life is chemistry. The other is the information paradigm, the idea that life is chemistry plus information. The discovery of the genetic code, on the other hand, has brought to light a 3rd key component of life, because a code is a set of rules that gives meaning to something, and meaning is different from both chemistry and information. In the Morse code, for example, the rule that dot dash corresponds to the letter A is equivalent to say that letter A is a meaning of dot dash. In the same way, the rule that a codon corresponds to an amino acid is equivalent to say that that amino acid is the meaning of that codon. Meaning, in other words, is a mental entity when the code is between mental objects, but it is an organic entity when the code is between organic molecules. This is why the discovery of the genetic code suggested a third solution, a third paradigm. The idea that life is chemistry, information, and meaning. But so far, this has always been a minority view. Despite the fact that meaning is the inevitable result of a code, modern biology has not accepted the existence of meaning in living systems, and this on the basis of two main arguments. The first argument was used to the stereochemical theory, the idea that the rules of the genetic code have been determined by stereochemical interactions between codons and amino acids. The outstanding consequence of this theory is that the genetic code is not a real code, because a code is real only when its rules are arbitrary. The rules of the Morse code, for example, are arbitrary because any letter of the alphabet could have been associated with any combination of dots and dashes. In the same way, the rules of the genetic code would be arbitrary if any amino acid could have been associated with any codon. And this is precisely what the stereochemical theory is denying. The second reason for rejecting the existence of meaning in living systems is the idea that there are only two types of codes in nature. The genetic code that appeared at the beginning of life and the codes of culture that arrived almost 4 billion years later. This amounts to saying that codes and meaning are not normal components of life because they are extraordinary exceptions that appeared only at the beginning and at the end of the history of life. These two arguments are still used to maintain that life is about chemistry and information, not about meaning. So it is important to show that all of them are in complete contrast with the experimental evidence. The first point, is the genetic code made of arbitrary rules or not? In protein synthesis, a sequence of codon is translated into a sequence of amino acids, and the bridge between them is realized by a third type of molecules called adapters. The adapters of protein synthesis are the transfer RNAs, molecules that at one side recognize a codon and at the other side associate it to an amino acid. The structure of the transfer RNAs clearly reveals that codons and amino acids are separated by a substantial molecular distance. And this suggests that a modification of the intermediate molecules could associate a different amino acid to the same codon, thus generating a different rules of the genetic code. And this is precisely what has been found in many laboratories ever since the classic experiment by Paul Simmel and colleagues. In 1988, for example, Hu and Simmel introduced two extra nucleotides into a transfer RNA without changing its anticodon and found that the resulting RNA was carrying a different amino acid. They had changed one of the rules of the genetic code. These and many other experiments have proved that the rules of the genetic code can be modified in countless different ways by adding or subtracting a few molecules in the transfer RNAs. This means that at the beginning of the evolution of the code, the number of potential adapters was virtually unlimited, and the potentially unlimited number of coding rules means, as in the Morse code, that they are arbitrary rules. The laboratory experiments, in short, have proved that the genetic code is made of arbitrary rules, and it is therefore a real code. Now, what about the second point? The fact that the genetic code was or was not an isolated exception. Well, many independent discoveries have shown that the genetic code was only the first of a long sequence of organic codes that have appeared in the history of life. Among them, the metabolic code, the sequence codes, the sugar code, the signal transduction codes, the splicing codes, the tubulin codes, the ubiquitin codes, the molecular codes, and the lamin codes. For the first 3 billion years of the history of life, the first 3,000 million years, evolution took place exclusively in single cells. But eventually some eukaryotic cell gave origin to multicellular creatures, to plants and animals, and new organic codes came into being. Among them, the Hox code, the adhesion code, the histone code, the transcriptional codes, the apoptosis code, and the bioelectric codes. With the origin of the nervous systems, a completely new type of codes came into being. Codes like the neural codes for mechanical stimuli, the neural codes for taste, the synaptic codes, the space code in the hippocampus, to which was given the 2014 Nobel Prize for Medicine, the acoustic codes, and the olfactory codes. According to recent estimates, the codes that have been discovered so far are more than 200. The organic codes that exist in living systems have appeared throughout the history of life, but their evolution has been very different in bacteria and in eukaryotes. It is an experimental fact that the number of organic codes in bacteria is much smaller than in eukaryotes. And it is also an experimental fact that the bacteria did not increase their complexity for millions of years, whereas the eukaryotes became increasingly more complex. They suggest that the bacteria did not increase their complexity because they did not evolve new codes, whereas the eukaryotes became increasingly more complex because they continued to evolve new codes. The complexity of a living system, in other words, appears to be closely associated with the number of its codes. This means that we need to re-examine the whole history of life, and in particular, the causes of that history. The processes that have become known as the mechanism of evolution. The mechanism of evolution have been one of the most controversial issues ever in biology. And the great debate about them culminated in the 1930s and 40s in the modern synthesis, or universal Darwinism. The theoretical framework according to which natural selection is the sole mechanism of evolution. Okay, but where does natural selection come from? We know that the copying of the genes is the elementary act that leads to heredity. But when the process of copying is repeated indefinitely, another phenomenon comes into being. Copying mistakes becomes inevitable, and in a world of limited resources, not all changes can survive, and the process of selection is bound to take place. Molecular copying, in other words, leads to heredity, and the indefinite repetition of molecular copying in a world of limited resources leads to natural selection. This is how natural selection came into existence. Molecular copying started it, and molecular copying has perpetuated it ever since, which amounts to saying that molecular selection would be the sole mechanism of evolution if molecular copying were the sole basic mechanism of life. The discovery of the genetic code, however, has proved that there are two molecular mechanisms at the basis of life: the copying of the genes and the coding of proteins. The discovery of other organic codes, furthermore, allows us to generalize this conclusion because it proves that coding is not limited to protein synthesis. Life, in other words, is not based on copying alone. It is based on copying and coding. And this means that evolution took place by two distinct mechanisms. More precisely, the existence of copying and coding at the molecular level means that there are two distinct types of evolutionary changes. Evolution by natural selection based on copying, and evolution by natural conventions based on coding. Now, the key point is that copying and coding are totally different mechanisms. Because copying is about information, whereas coding is about meaning. And this means that neither of them can be reduced to the other, and that both of them contributed to the history of life. The mechanism of copying and coding, furthermore, are both capable of bringing novelties into the world, but they do it in two very different ways. The copying mechanism produces either exact copies or slightly different versions of the copied molecules, which means that natural selection, the long-term results of molecular copying, generates new objects by modifying previous ones. Natural selection, in short, creates relative novelties, not absolute ones. In coding, instead, the situation is totally different because the rules of the code can establish relationships that have never existed before in the universe. Codes, in other words, can create absolute novelties, and this means that they've been responsible for the great absolute innovations that have appeared in the history of life. You also asked me about my experimental research, and that has been primarily dedicated to the study of ribosome microcrystals. A research that started in 1966 when I found that by cooling the eggs of one-day-old chick embryos at 4 degrees centigrade for 24 hours induced in their cells the formation of small ribosome microcrystals. Now, this discovery was important because getting the crystals of a molecule is the first step for reconstructing the three-dimensional structure of that molecule. The technology for reconstructing the structure of proteins from X-rays became available in the 1950s, and in the 1970s it was extended to viruses. Structures that are much more complex than proteins because they contain both nucleic acids and proteins and have molecular weights of about 1 million Dalton. They are very big molecules. The ribosomes are even more complex than viruses because in addition to heavier nucleic acid, they contain between 50 and 60 different proteins in prokaryotes, in bacteria, and between 70 and 80 proteins in eukaryotes. Organelles like nucleoli, mitochondria, or chloroplast are more complex than ribosomes, but they cannot form crystals because they are individually different from one another. They are supramolecular structures that do not have molecular identity, while ribosomes maintain this identity. This tells us that ribosomes have a unique place in nature. There is no other object in the universe that is more complex than ribosome and has the ability to crystallize. Eukaryotic ribosomes, furthermore, have molecular weights of about 4 million, while bacterial ribosomes are half the size and have weights of about 2 million. Now, the ribosomes of chick embryos are eukaryotic ribosomes. And what fascinated me is that their crystals have two extraordinary properties. They had the potential to reveal the three-dimensional structure of the ribosome, and at the same time, they were the most complex crystals in the universe. Okay? There is no other crystal which is more complex than the ribosome. Now, from the experimental point of view, the first problem was to obtain a significant amount of ribosome microcrystals. And to that purpose, I studied how to induce their formation in older embryos. This turned out to be possible, but only to a limited extent, because more differentiated cells produce less crystals. And in the end, I found that the best yield was obtained with 5-day-old chick embryos. In 1968, I discovered how to isolate ribosome microcrystals. And in 1970, Max Perus invited me to the Molecular Research Council in Cambridge, where I discussed the problem of obtaining information from those crystals, not only with Perus. Remember, Perus is the one who won the Nobel Prize for the structure of hemoglobin. Also, I discussed it with Francis Crick, Hugh Huxley, and Aaron Cook. Then from 1979 to 1980, I worked on ribosome crystallization at the Max Planck Institute for Molecular Genetics in Berlin. There, I discovered that the formation of microcrystals in chick embryos was actually due to biological factors, not to physical ones. More precisely, it was due to the fact that any damage to the ribosome transport system was halting the transport in a few places of the cells, and it was the accumulation of ribosomes in those small regions that induced them to form crystals. This led to me to the discovery that ribosome microcrystal can be formed not only in intact cells, but also in cell homogeneity. And it is actually in these systems that they reach their greatest dimensions. These results was published in 1979 in the Journal of Supramolecular Structure, and those microcrystals are still the biggest crystals of eukaryotic ribosomes that have been obtained so far. Now, all this experimental work has also a theoretical consequence. The fact that the formation of ribosome microcrystal depends upon the differentiation state of the cell raised a number of biological questions, and eventually these became evolutionary questions. What was the role of the ribosomes in evolution? Now, in 1909, Wilhelm Johansen proposed that the molecules that carry hereditary information, the genes, are different from the molecules that determine the structure of the cells, the proteins. And in order to distinguish the two categories, Johansen called them genotype and phenotype. At the beginning, this dualism was rejected because it was thought that proteins are responsible for both functions, heredity and structure. But in the 1950s, it became clear that only genes carry hereditary information, and the genotype-phenotype duality became universally accepted. On top of that, the computer gave an immediate legitimacy to the Johansen's duality, because the computer distinction between software and hardware is equivalent to the biological distinction between genotype and phenotype. And the cell was described as a biological computer made of biological software and biological hardware. Okay? Now, the problem with this model is that a computer does not generate its own codes. A computer receives the codes from a human operator, i.e. from an external agent, whereas the codes of the cell are generated inside the cells from an internal agent. This is why in 1981 I proposed that the cell is not a duality of genotype and phenotype, but a trinity of genotype, phenotype, and ribotype, where the ribotype is the ribonucleoprotein system of the cells that implements the rules of the genetic code. I proposed furthermore that the ribotype had an evolutionary priority over genotype and phenotype, because without the ribotype, without the genetic code, there is no correspondence between genes and proteins. So biological specificity could not exist before the genetic codes. There could be no genotype and phenotype before that. In 1985, I took a step further and proposed that evolution took place by two distinct mechanisms, by natural selections and by natural conventions. These are the two ideas of a new biology that I call semantic biology, i.e. biology with meaning, because it is based on the idea that codes and meaning are fundamental components of life. Now, then became a correspondence with Karl Popper and René Thom. Between 1981 and 1983, I had a correspondence with Karl Popper and with René Thom about these two ideas of semantic biology. The reaction of René Thom was immediately positive, and he offered me not only to publish my manuscript, but also to write the introduction of that book. The reaction of Karl Popper was more complex. He fully accepted the idea that the cell is a trinity, and actually he described it as a marvelous and as a revolutionary theory. But he was more cautious about the second idea, the idea that evolution took place by two distinct mechanisms. In reality, he was right in being cautious, because the idea that coding is a mechanism of evolution implies that many other codes appeared in the history of life, and in 1985 there was no evidence for that. It was only 20 years later that the existence of many organic codes became a reality and gave an experimental basis to the ideas of semantic biology. The introduction of meaning in biology was proposed not only by semantic biology or code biology, but also by biosemiotics. So let us see what is the difference between them. In May 2001, Thomas Sibiok invited me to review a special issue of Semiotica that was dedicated to the coming of age of biosemiotics and to celebrating Jakob von Uexküll as the chief architect of that new discipline. I accepted, and I acknowledged that the two main points of the special issue, the big one, is the making of biosemiotics and the recovery of Jakob von Uexküll from oblivion, came out with clarity and force and were a success. There was, however, a third point that I did not agree with. It was the idea that biosemiotics was the crowning achievement of the tradition that goes back to Goethe, Saint-Hilaire, von Baer, D'Arcy Thompson, and Drisch, a vitalistic tradition that has been the historical antagonist of the mechanistic approach of Galileo, Newton, Lamarck, Darwin, Mendel. I fully agreed with the project of introducing meaning in biology, but I argued that neo-vitalism was not the right approach. I sent my review to Sibiok in August 2001, saying that I had not been able to write an impartial report, and therefore that I would not be surprised if he turned it down. To my surprise, however, Sibiok accepted it. And to me, this meant that he was not against my approach. His implicit message, in my opinion, was that neo-vitalism is not compulsory. a mechanistic approach to the problem of meaning could not be ruled out, and people who were proposing it should be listened to. Seebeek died a few months later, on December 21, 2001, and that implicit message was probably his last contribution to biosemiotics. Personally, I took it as an invitation to join the biosemiotic community, and I decided to give it a try. The decisive change came in 2004 at the fourth gathering in biosemiotics organized by Anton Markos in Prague. Jesper Hofmeier, Klaus Hemmeken, Kalevi Kuhl, and Anton Markos and myself decided that what was uniting us, the introduction of meaning to biology, was far more important than our division, and we should make that visible. Up until then, I had been referring to the study of biological meaning as semantic biology, whereas Markos has been calling it biohermeneutics. But we accepted to give up our favorite names and to adopt the term biosemiotics that Seebeek has been campaigning for with so much passion and vigor. I also proposed to start a new journal, and a few years later I became the founder and the first editor-in-chief of the Springer Journal, Biosemiotics. Now, the problem is that, of course, things started to change. And started to change essentially because of the role of Charles Peirce in semiotics. Now, Charles Peirce was an American polymath who gave lasting contributions to philosophy, to logic, and to semiotics. In logic in particular, he was the first who went beyond Aristotle by adding the new category of abduction to the classical Aristotelian categories of induction and deduction. Abduction is an extrapolation from limited data, a mental jump to conclusion without enough data. Now, the Peircean concept of abduction and interpretation are important in biology because they have direct applications in the study of animal behavior. There is a beautiful example that illustrates this point. When a snake chases a prey and the prey hides behind the tree, the snake stops chasing. When a wolf chases a prey and the prey hides behind the tree, the wolf goes on chasing. The snake is only used codified rules, whereas the wolf is adding interpretation to them. The wolf is making a mental jump beyond the appearances, and that is what interpretation is, an abduction in the Peircean sense. An act of interpretation, on the other hand, is a process that gives meaning to something, and it is therefore by definition an act of semiosis. More precisely, it is a form of semiosis that is different from coding, because it is based on abductions and not on coding rules. So, we're realizing in this way that the Peircean concept of interpretation and abduction have a real explicative power, not only in semiotics, but also in biology, and are therefore their true scientific concept. Unfortunately, Thomas Seebeek and his followers came to the conclusion that interpretation takes place not only in the brains, but also in all living systems, including single cells. In reality, single cells do not build internal representations of the world, and therefore they cannot interpret them. There are simply no experimental evidence that interpretation takes place in the cell. And yet, that is precisely what the Peircean biosemiotics were claiming. More than that, they insisted that anyone who does not accept that the cell is capable of interpretation is automatically outside biosemiotics. And in 2012, I was expelled from the governing board of the Biosemiotics Society. That is how I lost my battle to turn biosemiotics into a scientific discipline. But I did not give up that battle. In the same year of my expulsion, I resigned as editor-in-chief of Biosemiotics and founded the new research field of code biology, a field that was explicitly defined in its constitution as the study of all codes of life, from the genetic code to the codes of culture, with a standard method of science. Okay? Now, there is a difference between code biology and many other theories which have introduced the concept of meaning in biology. So let us see what this difference is. You know, the idea that life requires not only information but also meaning was proposed for the first time in the semantic theory of evolution in 1985. The new biology proposed in that book was called Semantic Biology, a biology with meaning. But in 2012, after the discovery that many organic codes exist in living systems, that name was changed into code biology, because it is primarily the existence of codes that reveals the existence of meaning in living systems. That's the trick of it. The name was chosen because it is in codes that we have the experimental proof of the existence of meaning. And that justifies, in my opinion, the change of names. So from semantic biology, it becomes code biology. Now, code biology is based on an idea that set it apart from four major theoretical frameworks. Now, first, code biology is different from the modern synthesis because it claims that evolution took place not only by natural selection, but also by natural conventions. In addition to that, code biology describes the cell not as a biological computer made of genotype and phenotype, but as a trinity of genotype, phenotype, and ribotype, where the ribotype is the code maker of the cells, the ribonucleoprotein system that generates the rules of the genetic code. The second difference, code biology is different from the view that autopoiesis is the most fundamental process of life. Now, autopoiesis requires biological specificity, and specificity comes from the genetic code. So the ancestral system that came before that code could not have been autopoietic systems. Those ancestral systems, on the other hand, were engaged in the evolution of the genetic code and were therefore code-poietic systems. According to code biology, in other words, the most fundamental process of life is code-poiesis, not autopoiesis. The third difference, code biology is different from biosemiotics because it maintains that the process of interpretation takes place in the brain, but not in the cell. The fourth difference, code biology is different from the relational biology of Robert Rosen because it maintains that the process of anticipation takes place in the brain, but not in the cell. Code biology, in conclusion, started in 1985 with the idea that natural selection and natural conventions are two distinct mechanisms of evolution, and was officially established in 2012 when the International Society of Code Biology was founded. As a summary, it should be underlined that there are four major ideas in code biology. The first idea is that evolution took place by natural selection and by natural convention. There are two distinct mechanisms in evolution, not just one. The second idea is that the cell is not a biological computer. The cell is a trinity of genotype, phenotype, and ribotype. The cell contains the system that builds its own code. The third idea is that the complexity of a living system is determined by the number of its codes. That's what complexity is about. It's about codes, nothing else. The fourth system is that the fundamental process of life is code poiesis, not autopoiesis. Now, you also asked me about the difference between life and matter, between life and chemistry. And well, this is one of the most important issues in biology. So let us discuss it in 3 stages. The first stage is the difference that exists between the molecules of the living system and those of the inorganic world. Okay? Let's start from there. Is there a difference between the molecules of life and the molecules of chemistry? Now, let us start from the fact that all inorganic molecules are produced by spontaneous processes, whereas genes and proteins are produced by manufacturing processes. They are manufactured then by molecular machines that physically sticks their components in the order provided by templates. You know, genes and proteins are assembled by molecular robots, and this makes them as different from chemical molecules as artificial objects are from natural objects. Indeed, if we accept the traditional view that objects are natural when their structure is determined from within, and artificial when it is determined from without, we must conclude that genes and proteins are artificial molecules because they are determined from without. They are determined from external templates. They are artifacts manufactured by molecular machines. And this implies that all biological objects are artifacts. It implies that the whole of life is artifact making. Spontaneous genes and spontaneous proteins did appear on the primitive Earth, but they could not evolve into living systems because they do not have biological specificity. What they did instead was to produce molecular machines, and it was these machines that evolved into living systems. The simplest molecular machines that we can think of are molecules that can join other molecules together by chemical bonds. And for this reason, we can call them bond makers. Some could form bonds between amino acids, some between nucleotides, others between sugars, and so on. Among the various types of bond makers, some developed the ability to join nucleotides together in the order provided by a template. These bond makers started making copies of nucleic acid, so we can call them copy makers. Proteins, on the other hand, cannot be made by copying, and yet the information to make them must come from molecules that can be copied, because only these molecules can be inherited. So the information for manufacturing proteins must come from genes. So it was necessary to bring together a carrier of genetic information, a messenger RNA, a peptide bond maker, a piece of ribosomal RNA, and molecules that could associate codons to amino acid, transfer RNAs. The first protein makers, in other words, were code and template dependent peptide makers, or more simply, code makers. Life, in conclusion, appeared on the primitive Earth when the first molecular machines came into being and started populating our planet with manufactured molecules with molecular artifacts. Now, in order to discuss the divide between life and matter, it is useful to discuss first the divide that exists in physics between the great forces of nature: gravitation, electromagnetism, nuclear forces, and so forth. Now, the discoveries of science take place in two steps. First, we look at the world and choose a number of entities that describes it, entities that are known as observables, big space, time, mass, and so forth, precisely because they represent what we observe in nature. Then we look for relationship between the observables and obtain rules, regularities, and what we call the laws of nature. Now, in Newton's time, gravitation was the only universal force. But then electromagnetism was discovered and the universal forces became two. The difference between them is due to the fact that electromagnetism requires a new fundamental observable, the electric charge. And this is also true for the nuclear forces, for the elementary particles. We discover a new world only when we discover a new fundamental observable. Right? So, all of which raises the question, are there new observables in life? In reality, this is a rhetorical question because the idea that there are new observables is not an hypothesis, it is an experimental fact. We cannot describe living systems without genetic sequences, organic information, and without the genetic code, organic meaning. So these are observables because they are there, and we cannot describe living systems without them. But what kind of observables are they? In physics and chemistry, there are two types of observables. A small group of fundamental observables, for example, space, time, mass, charge, and temperature, and a much larger group of derived observables, the velocity, the forcing, etc. We need, therefore, to find out whether biological information belongs to the first or to the second group. Luckily, this problem has a straightforward solution because the sequences of genes and proteins have a very special characteristic. This is the fact that from the knowledge of n elements of a biological sequence, we cannot predict the element n plus 1. This is equivalent to saying that a specific sequence cannot be described by anything simpler than itself. So, it is necessarily a fundamental observable, not a derived one. The discovery of gravitation, of electromagnetism, of the nuclear force, etc., were all based on the discovery of new fundamental observables. And now we realize that this is also true in biology. Life is different from chemistry in the same way that electromagnetism is different from gravitation, because it requires new fundamental observables. Okay? Yeah, so the difference is there. But how real, how big, how substantial is difference? The philosophers say, is it an ontological difference or not? And the problem is this. The divide between life and matter does not consist only in the difference between their molecules. It is, first of all, an ontological problem. Is there an ontological difference between life and matter? We know that the sequence of nucleotide carries organic information, and that the rules of the genetic code create organic meaning. But what is the nature of information and meaning? What kind of entities are they from the ontological point of view? This is the problem of ontology. Now, according to a long tradition, natural entities are divided into quantities and qualities. Quantities are objective and can be measured, whereas qualities are subjective and cannot be measured. Okay? In the case of information and meaning, however, this scheme breaks down. Organic information is not a quantity because a specific sequence cannot be measured, but it's not a quality either because a specific sequence is an objective feature, not a subjective one. Now, the same is true for organic meaning. It is not a quantity because the rules of the genetic code cannot be measured, but it's not a quality either because they are objective rules, not subjective ones. So a scheme based on quantities and qualities alone is not enough to describe living systems. In addition to quantities that are objective and measurable, and to qualities which are subjective and not measurable, we must recognize the existence in life of a third type of entity, which are objective but not measurable. Organic information and organic meaning belongs precisely to the third types of entity. And since they can be described only by naming their components, we can say that they are nominable entities. They can be described only by naming their components. Now, another important point is that the structure of all inorganic molecules is computable because it can be calculated from its component, whereas the structure of the instance protein is non-computable because it can only be described by naming its component. Organic information, organic meaning inclusion are non-computable observables, and this makes them ontologically different from the observables of physics and chemistry. This is the ontological difference that exists between life and matter. Physics and chemistry are based on computable observables, whereas life is based on non-computable observables. Okay, that's it. That's the divide between life and matter, right? Thank you.